Good morning. I'm Alan Kornberg, Chair of the Board, and I want to welcome everyone, members, trustees, faculty, staff, supporters, to the 124th annual meeting. That's quite an accomplishment. First, let me apologize for not being there in person. 2.5 years, but got to Maine and unfortunately tested positive. So greetings from over here at Castine. Sorry, I can't be with you in Salisbury Cove. We have a very full agenda today, including some important and exciting updates from Herman Haller, president of the lab, and his colleagues. And those remarks are going to focus on the really exciting research underway on campus, as well as an update on the exciting and transformative plans we have for the future. So after we address the business parts of the meeting, we will turn to the more interesting part, which is what the future looks like and what's been going on currently. So with that, I'm going to call the meeting to order. I understand they were provided electronically to everyone who registered for today's meeting, and I believe they're also available in hard copy for those of you attending in person. So can I have a motion to approve the minutes? And I think someone in the auditorium is going to have to guide me as to whether or not we get motions and seconds. So will someone move for approval of the minutes? Yeah, I can't hear, so.
Someone motioned.
Okay, and is there a second?
Yes.
Great. Okay, now we're going to turn to the committee reports. The secretary's report, and I will turn it over to Jan Coates.
Can you hear me if I don't use a microphone?
Jan, if you could just get a little closer to the microphone, that would be great.
Okay. How's that?
Better, thank you.
Well, I'm pleased to give the secretary's report for this year. Incorporation membership now totals 237. It breaks out in the following ways. For full members, we have 4. For associate members, 5. Honorary members, 2. And life full members, 162. And life associate members, 36. Sadly, we lost 6 members who passed away this year. Life full member, Frank Moyer. Life associate member, Leslie Brewer and Robert Sayre. Full member and former trustees, Edward Clunan and Richard Crawford. Full member, Hugo DeYoung. And that concludes my report.
Okay, I think we a moment of silence to honor those that passed away. So if we could do that, I appreciate it. Okay, can we have a motion to approve the secretary?
So moved. So moved.
Second?
Second.
Thank you.
I think what Alan was trying to say is that Ed Vance is going to present the nominee. And since I'm Ed Vance, I'm going to go right ahead. So I'm pleased to report on the activities of the nominating committee. We are recommending to you the following slate of nominees for election to the Board of Trustees. This will be the class of 2026. For reappointments, Mr. John Hayes, Dr. Herman Holler, and Alan Miller, Esquire. And also, we're recommending a new member for the class of 2026, Ms. Anna Maynard. And if I could get a motion to approve. So moved. Second. All in favor? Motion carries. Also, we're pleased to present the following slate of nominees, which are submitted for membership on the 2023 nominating committee. That would be Edward Benz, MD as chair, Barbara Belts, PhD, Jerry Bowers, Claire Stone, Wistar Morris, Dr. Patricio Silva, and Daphne Trotter, Esquire. Could I get a motion to approve? So moved. All in favor? So the motion carries. Thank you very much. That's the nominating committee report.
Okay, next we have the treasurer. Going to
Thank you, Alan. Thanks, Herman. I'm Tom Boyd. I'll deliver the treasurer's reports. Could I have the next slide, please? Advance. Do this. Thank you. Next slide, please. So this slide summarizes the financial position of the laboratory as of the end of 2021 in comparison with the prior year, 2020. As you can see reflected in the numbers on the first line, the cash position of the laboratory remains strong. Cash, including certificates of deposit, was increased at 2021 year end by over $2 million compared with the previous year. This increase in total cash was associated with corresponding increases or decreases in the following areas. First, grants receivable increased by over $79,000 from year end 2020. This is included in the second line, receivable. Second, assets held in investment type accounts increased by $2.9 million over year end 2020. This is shown in the third line, investments. Third, the category called other liabilities decreased by $318,000 from year end 2020. That's the seventh line. In the category of bonds payable, the institution continues to pay down the $5 million bond that helped finance the Morris Laboratory building in 2007. We make regular principal and interest payments. $3.5 million in debt remains to be paid off, and there's a schedule for doing this by the year 2036. Total net assets are shown in the red box. Total net assets encompass assets with donor restrictions and assets without donor restrictions. Net assets amounted to $29 million at year end 2021, an increase of over $4.9 million from the prior year. Next slide, please. The $29 million in net assets are distributed among 46 different categories or funds. We go back to that. There we go. The largest category is plant assets, shown in green, which is land and buildings. The more liquid general unrestricted dollars in the reserve funds are shown in blue and red, respectively. The general unrestricted funds, the reserve funds, and the plant assets do not have restrictions. Other funds may be restricted by the donor for specific purposes. The institution may use all or a portion of the investment income that these restricted funds earn. All are tracked and accounted for separately. The reserve funds increased $4.2 million from prior year due to the launch and execution of a capital campaign and the receipt of $4.7 million. Some of the funds were already spent on strategic plan initiatives in that year. In addition to the funds listed above, the Maine Community Foundation manages the Salisbury Cove Research Fund established by Tom Marin and the Marin Foundation. Funds are made available each year, and we thank them for their ongoing support of the MDI Biological Laboratory. Next slide, please. The slide shows the statement of activities for the year 2021. Activities include operating support and revenues, as well as operating expenses. And the numbers are broken out by those with or without donor restrictions. Total operating support and revenue in 2021 was $16.8 million. Total operating expenses were $13.7 million, resulting in a positive change in net assets resulting from operations of $3.1 million. The non-operating change in net assets was a positive change of $1.9 million in 2021. This was due mainly to positive returns in the investment portfolio, government grants for capital purposes, and also contributions and non-government grants for capital purposes. In summary, in 2021, total net assets from combined operating and non-operating activities increased by $4.9 million, resulting in a balance of $29 million at the end of 2021. Next slide, please. This slide breaks out the categories of operating expenses. Total operating expenses were $13.7 million in 2021. If we exclude $1.4 million in non-cash depreciation, then operating expenses net of that amounted to $12.3 million for the year. Of these operating expenses, 73% or $9 million was for research and educational programs at Md Ibl, and also for monetary distributions to other institutions throughout the state of Maine through the INBRI program. And this is shown in blue and red. 22% of the expenses were for management in general. These are the costs necessary for the operation of the organization and the INBRI program, such as administration, record keeping, accounting, budgeting, legal, insurance, and grant reporting and compliance. This is shown in purple. And 5% was for fundraising activities for the purposes of obtaining contributions to support the institution. Next slide, please. This chart illustrates how the institution covered the $12.3 million in operating expenses for 2021. 69% came in the form of federal grants, contracts, and subawards. These are the blue and green sectors together. Breaking that out, 46% of the funding that covered operating expenses went to the MDI Biological Laboratory, and that's shown in the blue sector. And over 23% or $2.7 million was distributed to colleges and universities within the state through the INBRI program. And INBRI stands for the Idea Networks of Biomedical Research Excellence. And this is shown in the red sector. The laboratory was awarded 2 loans under the federal Paycheck Protection Program related to COVID. These loans amounted to 15% of the operating expenses for 2021. This is shown in the green sector. Both loans have been forgiven, and that means we don't have to pay them back. As in previous years, we highlight the 2 federal grants from the National Institutes of Health that continue to provide major support to the institution. First, the INBRI grant brings $1.5 million to the institution and $2.3 million to subawards to other institutions annually. And 2021 was the third year of a 5-year award cycle. And second, the COBRE grant. And COBRE stands for Centers of Biomedical Research Excellence. This amounts to $2.2 million of support, mainly for faculty at the lab. In 2021 was year 4 of a 5-year award cycle. And the funding for INBRE and COBRE grants are included in the blue and red sectors here. Next slide. That concludes the treasurer's report for the annual meeting. I'd be happy to take any questions.
Okay, can we have a motion to approve the treasurer's report?
So moved.
Second?
Thank you.
Was there a second? Okay,
Second, yep.
great. So now we can move on to the audit committee report, and I'll turn it over to Daphne Trotter.
I'm Daphne Trotter. Can you all hear me? I'm pleased to give the Audit and Risk Committee report today. The MDI Biological Laboratory Audit and Risk Committee met with Annette Gould, the auditor from HMV LLC, on Tuesday, July 26, 2022 at 2:30 p.m. Ms. Gould discussed the audit, what was covered, and some of the examinations performed. In the auditor's opinion, dated June 13, 2022, the financial statements present fairly in all material respects the financial position of Mount Desert Island Biological Laboratory as of December 31st, 2021, and the changes to its net assets and cash flows for the year then ended in accordance with accounting principles generally accepted in the United States of America. Ms. Gould reported that this year's audit went very smoothly and that no changes had to be made in numbers presented by the laboratory. That's the committee's report.
Can we have a motion to approve the audit committee report? Second?
Thank you.
Okay, thanks very much. So I think that concludes the business portion of the meeting. Before I turn it over to Herman, I just want to say a few things, which is that obviously the last couple of years have been extraordinarily challenging, and I think it's a testament to the leadership of the lab that we've not only survived, but we've thrived, and in particular, undertaken a lot of very important steps to plan for the future. And I just want to thank Herman, Ian, and the staff for doing really extraordinary work under very difficult circumstances. So I'm going to turn the meeting over to Herman, who has a bunch of updates. We'll talk about the very ambitious and exciting 5-year plan that he's laid out, and I'm sure you're all eager to hear what he has to say. So let me turn it over to him.
Thank you very much, Alan. We miss you here. We are still experiencing the difficulties of COVID-19 on an electronic basis. For instance, the sounds you hear or you heard all the time were new people entering the meeting, which was actually very positive but irritating. I'd like to welcome all of you to Md Ibl, both here and on Zoom, especially the members of the corporation, the board of trustees, and all our friends of the lab, and especially the members of the scientific advisory board. If I can, I think I'll do this here. Roy, I think I need some help. Yeah, but I need to point as well, so So I don't know why. No, the battery is there.
There's a whiteboard behind you.
Yeah, you know that I'm very fond of white or blackboard. So I wanted to especially welcome the members of our scientific advisory board. And this is because we have our meeting with members of the scientific advisory board for the first time in a long time. We didn't have a meeting for the last 10 years, actually. And here you see the list. Not all of our members of the scientific advisory board are with us, but they are with us today. So for them, it will be an introduction to the lab. And Corinne, if you allow, you just gave me a very nice example of how necessary this is, because Corinne Antignac, who is here for the first time, turned to me and asked, "What's this picture?" And I said, "Well, that's Md Ibl." And she said, "Well, it looks much bigger on screen than it does in actual life." I can assure you it's much bigger in real life than on this screen. But we have to talk about that, to learn about that. Some of you know that although I'm not sailing, not an ex-sailor, next slide, I like to compare the lab to one of these ships. You know, elegant ships, 124 years old, you have just heard that. And we worked very hard to make this ship ready to sail. This does not tell you that we're there yet, but we have started our journey. Sometimes we think we are repairing this thing while it's sailing, which is true, but we try to prepare ourselves for the journey ahead of us, and this is what I'm going to talk about. So we achieved a lot, but I think you have to be aware that this ship has been used for a very long time only for summer trips. That's very different when you go out there during the main winter. You have really to rig the ship differently. You need a different crew, and this is what we have been trying to achieve, excuse me, over the last 10 years. And we worked very hard over the last 4 years to get ready to sail. These are some of the things we have been doing. Over the last 2 years, and I will not be able to address all of this. I'll go to some of these. We will not talk a lot about successful recruitment because we did this over the last 2 years. New research groups, more grant applications, the graduate program I'll talk about, the visiting scientist program, we'll talk about animal facilities and microscopy and new initiatives like MDI Biosciences. But before I go into all this, I think it is very important to realize that it was not easy, and it is still not easy. Alan Kornberg already mentioned this, but you all know when you're here on the island or wherever you are, your restaurants are not open all the time. Sometimes you have a reservation, but they are closing because of COVID, because of shortage of staff. And this is what we have experienced here, but we can't close down. We have our animals to take care of. We have an ongoing, we have work in the labs, and I'm very proud that we didn't shut down, not a bit. So our animal facilities were running despite we are spread thin to start with. You'll hear a story about success. We have achieved a lot, but our staff is still the same as it was 4 years ago. This means that everybody is working very hard and very enthusiastically, but then in addition, everything breaks down because we have post-COVID going from 0 to 95, but we don't have the staff for that. We have a difficult job market. We have difficult support lines. All the prices are going up. So we have survived, but we also have changes. Work-life balance after COVID is not the same as before. And we all know about that. And I will not talk about this any further, but you have to realize this is the background we're working with at the moment. And we only survive because of the group. For the last 2 years, everybody has working very hard. I'll give you one example. Animal facility, very important. These animals have to be fed and clean. We have exchanged the whole team. We had to exchange the whole team, Ian and all the others, 2 times over the last 2 years. So administration was trained how to feed zebrafish. Thankfully, they didn't have to do it. But nonetheless, you were there. And this is true for other areas as well. So I'd like to give a round of applause to all the people who have worked very hard. Especially the ones sitting over there. And when you talk to the scientists, we did much better than a lot of other institutions. Our research was going perhaps not 95, but at least 70. We were publishing, we were writing grants. You see all these successes, and this is on the background of everybody working very hard. So I'll talk about the crew, the ship, and the challenges. And I'd like to start first with this. Some of you have this in hand. Mine is over there. This is the map. You know, when you're sailing, you have to know in which direction you're going. And this is a very important map, and we have worked on that, and I'll talk along the lines of this map. And the text is important, and the numbers are important, because this is our strategic plan. And this is built on the financial situation of the lab, of what we need. And when I outline now what we are going to do and what we have achieved, this is what we need. So we are not naive. We know how hard this is, and we are optimistic. I think we can really get there. But first, let me show you where we are. So the group. This is Md Ibl. This is the science. These are the research groups working here, and tomorrow we'll sit down with the scientific advisory board, and all the different groups will present their work, and we'll have critique, support for what we are doing and in which direction we want to develop. I will not go through Ian's work. He'll talk about science in a moment. My work, James Godwin, Brian Muravala, Sam Beck, Romain Madeleine, Eric Rogers, Jared Rollins, Dustin Updike, Tim Kaufman, and Jane Disson. We have promoted 2 of our scientists. Dustin Updike is now associate professor at Md Ibl, and Jane Disson is associate professor of environmental health. So we still have the same focus, which is regeneration and aging, toxicology and environment. These are the overarching goals of Md Ibl. But the core of everything, and not only for the research, but also for the planning of Md Ibl, are the research groups and the structure of the research group. So one important question is how many research groups do you think Md Ibl should have? And this depends obviously on the quality. But when we think about the future, I think 12 to 14 research groups, that's the number which is good for MD Ibl. This is big enough so that we have a lot of interaction and we have a lot of communication, but it's still small enough to keep us focused. One of the role models we have are Max Planck Institute. That's a German model, something like Howard Hughes. And this is the size, 12 to 14 groups of a Max Planck group in Germany, a Max Planck Institute. And the reason why I think this is a good role model, because 90% of all the Nobel Prize winners in Germany are not coming out of universities, but coming out of Max Planck Institutes. So I think that's a pretty good role model for us. So this is the size we want to achieve. However, it depends a lot on what a research group actually is. You know, here you see the leaders of these research groups. Some of you have already noticed that we have a diversity problem. We know that, and we're working on that. But the real question is, what is a research group? And I'd like to give you 3 examples. Too many, I can't go through all of them, but to give you an idea what we understand, how a research group is formed, and then we have numbers in order to calculate research space and housing and a financial plan. So 3 examples. This is Dustin Updike's group. They are standing precautiously near to the water here, Dustin. You are a little bit safer, but all the others, you know, 1 step backwards, and here we go. So you see the list of postdocs in the lab, 2 postdocs, Dustin, research assistant, 2 summer students, and then there is a graduate student who will join the lab next year. So when you look at the numbers here, and we are not talking about the quality of research, Ian will do this, then we have 8, 9 people in this research group, which gives us a number. It may change. It may be that Dustin moves somewhere else because he gets a very attractive offer somewhere else. We have mixed feelings about that, you know. On the one hand, it would be a catastrophe. On the other hand, we would be very proud. But nonetheless, we have 2 summer students here. So these summer students will not be here in December, but they will come back next year. So we have to plan for them. The graduate student will stay in Dustin's group for around 4 years. Well, I know. So, but these are all the things we have to take into account. Next example is Prayak Muravala's group. Prayak arrived here 3 years ago, 2.5 years ago, 2 years ago. So this is a fast-growing group. Here you see Prayak, you see graduate students, postdocs, you see summer students, and then you have 2 more graduate students who will join the group. You may have noticed that in Dustin's group, we had 1 visiting scientist. Here we have now 4 visiting scientists. Some of them are here only for a couple of days, but some of them are staying for longer. In Dustin's case, the visiting scientist has been coming back now for 3 years, 4 years, 3 years. So when you count here, although they are not here all the time, the number is higher. And this is my research group. Looks huge. 6 summer students, which is a lot of work. I mean, some of them are sitting over there. It was very hard during this summer, but then they're gone, much less people. Corey Johnson, he is graduated yesterday, and he'll join the lab in October. And then we have a postdoc next spring from Germany, and these are visiting scientists. Now, this is difficult Vera came here for 4 weeks in June. She'll be back in September for another 6 weeks. Helmut Augustin is planning to send students, was here for altogether 2 weeks this year. Bert Fricker and Jörg Huwiler have been visiting scientists for a long time. They are now more integrated and they stay for a couple of weeks. Bernard is from Leiden University, has been here 7 years ago for a long summer. Now he wants to join the lab for a year together with his family. And on Dean Kleber, we are negotiating at the moment about next year and how she can be incorporated. So this is a large number and gives you an impression also about the high variability we have when we plan for that. I'll come to our campus in a moment because we have the possibility. Others like the Czechs, they are much more limited. So we are talking then perhaps more than 100, but definitely more than 50 people working in science at Md Ibl with a high fluctuation. And I think it's important for you to understand all this because otherwise it's just research group and research group, altogether 15. What it means is actually this fluctuation. That's the beauty of academia. You know, this is why we're doing that. It's lively. We have people coming in. But you can also understand that for staff, it's very difficult. Some of them, you are serious. Next week, somebody is coming for 6 weeks, staying on campus. Yes, we forgot to talk about that. But this is why we have the map. This is why we need more space. We need more supporting staff. We need more of most everything in order to keep this ship going. The first problem is, how do we get all these people here? So we have to reach, because we are tiny here, we have to reach out to the world, and we do this to other partner institutions. And the first level, very important, are graduate students. You can't get a degree at Md Ibl. So we have to reach out to other institutions. Not easy, because to be honest, they hate giving us their students. Well, it's okay for 6 weeks or perhaps 2 months so that they get an international experience somewhere else. But we are talking 4 years. We want to have all of them. We want to train them. We want them to be successful. So we have now our own graduate program, and the students come from different parts of the world. Kylie Bragdon is responsible for the program. She's actually not coming from far away. She's from Winter Harbor, which gives our students the experience of a PhD who actually knows how to handle a lobster boat, which I think is very special. Now, our students, we are very proud of them, but this means that our graduate program has to cover all the other graduate programs in order to fulfill the requirements for their degree. In fact, we have to be better than the other programs so that it's attractive to be here at Md Ibl. We are responsible for their career. If they don't publish well, if they don't have a career, I can tell you they will not tell their colleagues they should apply for a place at Md Ibl. We went to Germany because quite a few of our students are coming through the Hanover program. This is the meeting we had 13th of May in Hanover, and you can very nicely see that there is a lot of interaction, one German group, one Md Ibl group, and it worked. We have now Hanover researchers coming here. Alina Sharbata, who gave this talk, will be with us in a week. More collaborations have started out of that, but this tells you we are Maine, but we have to go out there and we have to collaborate and we have to interact with the world out there. We have postdocs coming from different parts of the world. We're used to that. All academic institutions have this, but they are financed both through our grants, but also through international grants from outside, and we have to work on that. And here you see our map. This is world-class education and training. That's what I just said. That's what we're going to do. I mean, it's world-class training. 6 million. After what we have calculated, this is what we need in order to keep these programs going, in order to support our educational program. We also, as you know, we have a visiting scientist program. This has changed also. We always had visitors here from all over the world, and this gives you a short list, and some of them you have already seen before when I showed the pictures of the different groups. We have a different structure now. When you are interested in becoming a visiting scientist, you either talk to a research group leader directly, or you apply to the lab, and then we have a seminar where all the faculty members listen in with discussion. And then they are invited to come here because they have to get to know Md Ibl. And then we try to find collaborations. We try to find where these visiting scientists would best fit in. Sometimes there is already a project, but sometimes not. And some of them want to come for longer periods or establish more long-lasting relationships. Now, space and equipment. I mean, the first problem was to understand how a research group functions, about our international and national collaborations and the fluctuations which we have. How do we manage this? And everybody should be happy, but it's tight. And you can still see we are on a ship. I mean, everybody has to find space. So first, our laboratories. We work, as you know, in Davis and in Morris, and we have the laboratories on the lower campus, and we have Karnowski. If we start, and if you are able, and we are planning to do this starting next year, we will renovate the Neil laboratory. The roof has to go up. We have to have more offices in there. We'll plan so that a lot of researchers can work in there. We'll have to renovate Karnowski. Not that much. My research group has been working there very efficiently for more than 10 years. But nonetheless, this is the lab space where we can increase at the moment. So we are not, I think this is important, we are at the moment not talking about a new lab building. I think this will be 2030 and afterwards. For the time being, 14 research groups with all the fluctuation you have seen, I think we can manage. However, our animals, we have the most wonderful animal facility I know. We have more non-mammalian animal models within this small institution than most academic institutions I know about. We have specialists in zebrafish. We are for kidney with Ian, the center of the world for kidney and zebrafish. We have now axolotl, and I still don't know whether we are the second or third largest axolotl facility in the US. Brian can change. So this tells you lots. And these wonderful animals, we are the center. We have a lot of transgenic animals. So we are, in a way, the repository for axolotl. We have wonderful groups working on C. elegans. We have introduced African turquoise killifish. And Eric has told me a couple of days ago that he's very proud that he gets now calls when people are asking us, and we consider ourselves newcomers in the field, how to work best with ATK. And we have stem cell derived organoids. We have lost Vicky Losick with Drosophila. She went to Boston. That's what happens in academia. But we hope that we can reestablish Drosophila with the help of Halina Scarpato. And this means space. This shows you the animal facilities down there in the basement. That's hard work. We already applauded the staff, but I can tell you the animal facility, outstanding job, because they provide the basis for our research. And here we have to expand. Here you see Ian standing in front of the gull shed. History, when I started here, this is where you could meet the sharks. But no sharks anymore, but more zebrafish and more axolotl and more ATK in the future. So this is one important core facility. And the second, we have more than these two, which is very important, is microscopy. Because when we think about the development of Md Ibl and what you actually can achieve here, what we want people to come here and to have a little bit more time to discuss things and to sit down and actually look at their microscopy pictures and discuss these. So we have been working very hard, and we applied and wrote grants, and Ian and the whole faculty worked very hard. So we have now the most outstanding microscopy facility in this part of the US. And I'm not talking about Maine, I'm talking about the East Coast. And this, you can see here some of the examples, what we can visualize now, so we can ask more specific questions. And this will be our latest achievement. This will be a mesospin microscope where we can visualize larger animals. This will be the only one, not in the US, but east of the Mississippi. And this will be assembled by ourselves with the help of external people. So I think Jerry has set up a time-lapse video in October, November, because it's IKEA at Md Ibl. We have around 500 screws and larger pieces, and we'll put this together. And I think it will be fun to watch these scientists. Where is this screw? Now it can't be. There were 5 of them, and so we're looking forward to this. But once it's working, people are already lining up to come to Md Ibl just to work with us on these microscopes.
Did they send the directions?
Sometimes from what I've seen and what I've heard, sometimes only the parts arrive and no description whatsoever. Because I've told you that it's the first microscope. So this is not that it's not being produced by somebody else. We assemble it. 11 million, supporting outstanding science. Everything I've been talking about for the last 10 minutes, this is what we need. Now I can see that I have to. Campus, very briefly. Our campus, as you know, is iconic. Corinne, this is all our campus, the whole thing. So basically, we are talking about a science campus, and we have grouped this. We are working on a strategic plan for the whole campus. And we have the Spruce Point area. We have areas where we plan professional housing. We have to think with the size you have just seen about more community research and administration. We have to improve. And the different areas, last not least, the pond area. And these are the plans for the next coming years. So we'll work on the Spruce Point area. Here in white, you see all the cottages we have still to winterize and renovate. So this is really big. We have started here in green. You see the ones we have renovated. And to be honest, if we wouldn't have started this right away 4 years ago, we would not have been able to house the Scientific Advisory Board or some of the other visitors coming this summer. But if we are serious about the program I've just told you, we need these renovations for the students. We need new professional housing, and we are planning for that. You can see in green the ones we have already renovated, and some need not that much renovation. But then something like the pond area, that's also big. This is the plan. We want to make use of our campus. And you all know, especially the ones who have been here during the last 20 years, we were always proud of our campus and the stories which go with the campus. I will not go into that, Ed, but I could quote you about killing more mice in Cove 3 than during your research at Harvard. And I love all these stories. But for the future, to be honest, it can't be like that. We have to be more modern, and we have to keep the beauty and the charm of Md Ibl despite being more modern. 11 million. So please have a look at the map. Then you know where we will sail, and you know which part of the journey you actually want to participate in. I'm almost done. Very briefly, unfortunately, about courses and conferences. That's not 2022. Dennis, you can see yourself, that was not this year. You've heard a lot about IMBRI already during the finance report. IMBRI is very important for the lab, was just renewed. It's not only important because of the money which comes into Md Ibl, but we are the center of biomedical education in the state. And one of the things Jim Kaufman, who is responsible for IMBRI at the moment, and I will do during the winter, to travel to all these institutions and to revitalize our relationship with them and discuss. Chris Smith and the whole crew is doing a great job in professional development. And we have talked about some of these courses and the symposia. You see African turquoise killifish, biomedical innovation. With the microscopy I've just described, we have to communicate this not only by publications, but by having courses and conferences. And then we also have undergraduates here, very important, because this is the next generation. And I'm very proud of all the students who are coming back. And I'm very proud of students who actually return to Md Ibl after graduation. And so this is a very important part. I already mentioned this. It's very important in the future that we have our own, what was called signature courses at Md Ibl, which are faculty-run, faculty-initiated courses where we educate the world in the ICAR course that's on aging and regeneration. The microscopy course has started this year. Biotechnology and drug development also started this year together with Covey College. Branches from the same tree, art meets science. I'm very fond of that. And then you can see what's in the plan. Unfortunately, I don't have time for that. 18th, 19th of August, if you're seriously interested in 2 branches from the same tree, what this means, please join them. Md Ibl within Maine, very important topic. Although we have been in Maine for 124 years, for a long time, Maine didn't realize that Md Ibl really existed. because MD Ibl was the place where all these researchers came from far away, and then they stayed for some time, and then they went back. This does not mean that we are not connected to Maine, but we are not connected in the way we should be. When you look at MBRI, we are responsible for biomedical education, and we are an important partner. And 49th Maine Biomedical Science Symposium this year at MD Ibl, very important. But we have started to work now, and we have more collaborations. I already mentioned a course together with Colby and the president of Colby, and they will come, the faculty, later in the fall. We just heard that the Rue Institute, new institution in Portland, has applied for a very large NSF grant, and we are part of that. We are an integral part of this new application. A new COPRI most likely will be funded, and Romain Madeleine from MD Ibl and Ian Drummond are part of that. And then we are also collaborating much more with the University of Maine. Some of us have now joint professorships there. We are sitting on their committees. So MD Ibl is much more integrated within the Maine institution. And in October this year, there will be the first annual statewide research symposium. Jim Kaufman will complain and will say, well, you've just heard that the 48th symposium was at MD Ibl, but this is really statewide and will address the NIH. So NIH leadership has already agreed to come. So this will be a presentation of our institutions, and we are a big part in the planning. We play a big part in the planning of this. So ready to sail. We will not sink. And we know where we want to go to, but we need a lot of support for that. We need support from you. We need critical support. We can do this together. I will not read this out. This is our vision of MD Ibl, which is still true. It's 4 years old. We have a wonderful institution. We have now an outstanding faculty and all the resources we need to be part of international science. And we have the most wonderful campus on the planet. Well, perhaps not on the planet, but it's a wonderful campus. And with that, I'll hand over to the last part of our map. And this is Jim Strickland. I mean, Jim, I can't help you. We are both responsible for that. But now you have to tell us about this here. And this is our last initiative, MDI Bioscience. And this is the Halsey Lab, which is now MDI Biosciences. And some of you may remember that. This is also the Halsey Lab, but from a different angle. I can tell you when I gave my first talk here, I think it was in 2001 or so, I was shitting my pants. And this is what we are changing. We still have the Halsey Lab, but within the Halsey Lab, and Jim will tell you about that, it will be a very, very modern lab. So keeping the history and keeping the tradition, and at the same time have to have the best scientific institution, that's what we are aiming for. And with that, Jim, it's all yours. And I hope I can start your slides from here. Or Roy has to help. Thank you very much for your attention. Yes, I was expecting that.
I've always assumed that the reason we didn't have a graduate was that we didn't have a football. But it seems like we have so many academic institutions in our network, and we're now in an age where so much of education occurs remotely. It doesn't seem to be conceptually difficult to think of us having.
Yeah. Well, it was to be expected, a question which I can't answer in one sentence. We'll talk about this during lunch. First of all, if we go into sports, it will be hockey. I can't help it. We have at the moment several people and they play hockey. So a hockey team is the most likely team we'll have. And secondly, yes, remote, very necessary. But the beauty of this campus, the experience of being at Md Ibl and doing research here, to be honest, I don't think we can replace this.
No, it's the other part that needs replaced.
Okay. Let's talk about it over lunch. Tim, it's all yours.
Can you hear me okay? There we go. Perfect. Well, my name is Jim Strickland. I'm the director of MDI Bioscience, the new initiative here at the lab. And I first just wanted to start out and we've talked about StarPoint in this beautiful slide this morning already, but for me, being a Midwesterner, I still walk out into StarPoint and I pinch myself. What an incredible place to be. And what an incredible history that this lab has as a mecca really for comparative biology. And I'm a big student of the history of the lab, and I'm in awe of being a part of that history and launching MDI Bioscience, which may feel like a new initiative, but I would argue it's actually more of an extension of the work that has been done for 124 years here at the lab, as we now transition to using comparative biology to explore how we can impact the development of new pharmaceuticals. So for those who aren't familiar with the pharmaceutical development process, it is a daunting pathway. And you've all probably tired of the statistic of 1 in 5,000 to 10,000 compounds actually make it to market. But it's true. It's an incredible attrition rate. And the fact that there's a business model at the end of this, that you can have less than 1% success rate and still have a business, is rather remarkable in its own right. So when you look at this continuum, you can see that from discovery through preclinical animal studies to clinical trials to FDA approval, there is just an incredible weaning of compounds, and we think we can play a role in helping make that more efficient. $2.5 billion to get a drug to market in 15 years. And I think as a society, we all want cheaper drugs, and a way to do that would be to make this process much more efficient. So as we think about in specific where MDI Bioscience and Md Ibl can play a role, the goal of getting to a first human clinical study is always a bit of a daunting one. And companies spend a mean of 161 million to get from discovery into a first human clinical study. And we think that better characterizing new compounds and helping understand the innate properties of their efficacy and toxicity earlier in development will play a significant role in making that process more efficient. So why Md Ibl? Well, we've already established that comparative biology is a wonderful tool in assessing disease conditions for basic research. And I would argue, and I think it's well supported by the work and the literature that has come out over the last decade or so, that zebrafish and non-mammalian models offer tremendous potential to translate the work in basic science into new pharmaceutical development pathways. So MDI Bioscience is really an extension of the tools and the wonderful expertise here at Md Ibl. And what we're seeking to do is essentially better informed decision-making so that pharmaceutical companies can increase the rate of translation from, in particular, preclinical to clinical stage compounds And we're going to do that really using three different methods. One is zebrafish and other non-mammalian models offer a tremendous opportunity to study disease at the molecular level. And so developing disease models that help us emulate or induce disease in animal models and study that those diseases and their mechanisms is a really unique opportunity for the lab. And secondary to that is once you have the disease model developed, it gives you a wonderful platform then to screen compounds, large compound libraries, to evaluate whether or not you're able to see a signal of efficacy where you can restore function, or in the inverse, if you're looking at the potential for toxicity, you want to find that out as early as possible as well. And then lastly, but certainly not least, is the use of bioinformatics and computational pharmacology as a way to improve the predictivity of the non-mammalian models and elucidating efficacy and toxicity. So zebrafish are sort of our lead model for drug discovery, and there's good reason for that. The publication of studies of zebrafish in drug discovery over the last 10 to 15 years has skyrocketed. And so while mammalian models still remain critical to getting drugs to market, there's a unique role that a zebrafish can play in drug discovery, in that it's extremely genetically similar to humans, and 82% of the genes that cause disease in humans are preserved in a zebrafish. And then structurally, they're very similar from an anatomical and cellular perspective. And then last but not least, and this offers a tremendous benefit over mammalian systems, the small, transparent zebrafish embryo is a wonderful platform for high throughput screening, where you can look at mass numbers of compounds very time and cost effectively and discover whether or not they're effective against a particular disease model or might offer toxicity. So when you look at the continuum of drug discovery, we play at the very leading edge, where compounds are really early in sort of assessing the potential viability of a compound and its ability to restore function, or its characterization of potential toxicity. And certainly later on, the mammalian models play the primary role in essentially assessing toxicity and safety prior to actual introduction into human clinical studies. But zebrafish has a really unique opportunity to play in that early discovery phase, where you're looking at mechanisms and trying to identify which compound has the most potential going forward. On the left here, you have sort of typical drug discovery today. Right now, everything focuses on isolated cell systems. And while that is convenient and it's offers the potential to evaluate large numbers of compounds easily and cost effectively, we know that isolated cells lack native function. They contain mutations and they may be impure. So there are some weaknesses to using cell biology as an indicator of efficacy and toxicity. And so our goal is to transform that model to one that integrates in vivo biology, whole systems biology, where you can actually assess efficacy and toxicity, both in cell culture as well as in an intact biological organism early in the process, so that you can reduce the amount of time it takes to get rid of compounds that have a negative profile and accelerate the compounds that have the properties that you're looking for and have a great potential to address disease. And that enables staging of your non-clinical work and mammalian studies much more efficiently as well. So how does MDI Bioscience engage with other organizations and pharmaceutical industry partners in general? We're really working under a problem-specific approach where we're consulting with pharmaceutical companies to better understand what problem is that they're looking to, what the problem is that they're looking to solve, and what mechanism of injury they want to induce, and how to best restore function using that disease model. And so once we are able to essentially develop the model that suits their particular needs, we can, or identify the research goals, we can then enlist MDI scientists and faculty to help with addressing that research problem, as well as external collaborators such as visiting scientists and our scientific advisory board. And then, as Dr. Haller mentioned, we're renovating the Halsey lab so it can be a really top-notch facility for pharmaceutical research, and that requires complying with industry standards with regard to operating procedures, security, and good laboratory practices. So this is not a small undertaking, and it's one that we're in the middle of doing right now, and I think it's going to pay off in the long term. So for the immediate future, we're building the blueprint for MDI Bioscience at the moment. We've spent a lot of time with industry this year, having many conversations, understanding what they need from their development models, and then also educating them. This is not something, the use of zebrafish is not something that's routinely done. So we're educating the market as we go along as well. But we found that there's an appetite. We found that there's an appetite for innovation and for new ways of thinking in early stage drug discovery, and that's been refreshing. We also have heard that in addition to that customized approach to developing disease models and screening new compounds, that there's also a desire to have some toxicology models that can be more of an off-the-shelf type of product that allows companies to essentially pressure test compounds against some in vivo models of toxicology. And so we have a plan to go about developing those screens. And we expect that in early 2023, we'll have fish bred and ready to go in the Halsey facility so that we're ready to accept studies and have pharmaceutical audits of the facility and be ready to undertake work. That's great because we have several late stage discussions right now with pharmaceutical companies where they're hopefully, we hope we're going to have 1 or 2 signed contracts yet this year where we can go ahead and have business ready to go for when the Halsey lab is fully renovated. And that's also matched by a robust fall meeting schedule where we have many different professional societies and meetings where we will be actually speaking about the use of zebrafish. So we'll have the opportunity to continue to develop the business pipeline as we go through the fall. Above all, I think the last message is just this is early stage. So we're being opportunistic and flexible and understanding that we don't yet have all the answers. So we're working through this and making sure that we're being thoughtful in how we receive feedback and how we solicit feedback and how we then use that to build MDI Bioscience. And then also we've had so many new opportunities come at us that we're also trying to work through opportunities to build partnerships and build the potential for MDI bioscience in a thoughtful and strategic way. And with that, thank you for the opportunity.
Thank you very much, Tim. For those of you who are interested, how will this develop? We have two examples here. Either this will develop into an independent company, but the tax has the same model for mice, and they are still a nonprofit. So we are talking at the moment, we are discussing this with lawyers, how this will develop. First, it's a great pleasure to do this together with Jim. I mean, the whole faculty is very supportive, and he's combining the focus of an entrepreneur, because that's what you need, with the openness of a scientist. I could turn this around, the focus of a scientist and the openness of an entrepreneur. And nonetheless, it's a pleasure to do this together. And now, it's a little bit like Christmas, Ian. We have been waiting for you all the time, because he will talk about what's really important about the research, what we have been doing, what we have achieved over the last 3 years. Yes, I wouldn't say that, but we haven't talked about the boat so far, but
Suffice it to say, we know the rocks are there and damn. My job is to tell you about our achievements over the last year, which have been strong. We've had a very strong year despite usual COVID issues. I think we've been very productive. We might actually on.
Okay. Good. I'll tell you a bit about our ongoing program of grant submissions to keep the science funded and rolling forward, and exciting new developments you've heard some about with the microscopy innovations we're working on here. So we've had 23 publications over the last year, and I'll run through a representative bunch of them, not all of them, to highlight sort of the impact we think we're having here at Md Ibl, and how we're keeping ourselves at the forefront of aging and regeneration. So Dustin Updike's group published a nice paper on the C. elegans germ cells, and his group is essentially defining the way that germ cells maintain their potential to become all cells of the organism, pluripotency. So he's looking at these very specific structures within cells that may mediate the preservation of this pluripotency. But more than that, his work is addressing a new way of organizing the cytoplasm. Basically, how machinery in the cytoplasm is organized without membranes around it, without obvious structure, more in a self-assembly process, to be little factories for a process in the cell that I think we'll see a lot more of in the next 10 years. So Dustin is a leader in that. Eric Rogers' group continues at the interface of metabolism and aging and metabolism and lifespan extension, and is doing some very interesting work defining targets for intervention in lifespan extension. My own group takes a lead role in this NIH consortium about rebuilding kidneys, and I published this along with all the others. And Dennis is on the advisory board for that consortium as well. So we're innovating in using the fish as a model of kidney regeneration, because it does regenerate its kidney. We're trying to figure out how. Please and thank you. I think we have a little glitch. Oh, hang on. It's probably me. Let's try that. There we go. James Godwin is doing some wonderful work in limb regeneration, and more importantly, defining what are the parameters that allow limbs to regenerate, for instance, in the axolotl, but less well in mammals, and really define the different cell types, different origins of cells that contribute to regeneration. And so that has obvious implications for wound healing and regenerating our any amputated appendages. Another paper from Eric, a collaborative paper looking at autophagy, which is the process of basically recycling the proteins within your cell to repurpose them and preserve function for a longer lifespan. One of our mutants turned out not to make any females. So surprise, surprise, we defined a new gene required for gonad development in the fish with obvious implications for higher organisms. Again, James's group is looking at immune function in regeneration and defining how the axolotl does it differently to allow for a regenerative response instead of a fibrotic scarring response. And so his work is advancing nicely towards defining the similarities, but more importantly, the very differences, the bifurcation points between organisms so that we can learn what to tweak in humans to promote regeneration. Prayag Mirawala is taking a leadership role internationally in defining the axolotl genome, the nomenclature for all the genes, tools for analyzing it. And this is essential for any model organism. And of course, here at MD Ibl, we pride ourselves on all our non-mammalian model organisms. And the axolotl, with his work, introducing new transgenic approaches to really modernize the system, he's taking a lead role in defining how we can work better with the axolotl. Jim Kaufman is leading the way in understanding early stress responses, genetic regulatory circuits, and how they impact the organism long-term. Doing very refined work on glucocorticoid receptors and basically how targets for the glucocorticoid receptor and what's activated by steroid or by cortisol has intricate feedback mechanisms that promote and sustain signaling, perhaps dysfunctionally, after stress. A major paper for Sam Beck's group was looking at how gene regulation is basically noisy as we age, and that misexpression of genes can have pathological impacts and be part of an aging pathology. But more than just defining this with this very powerful overarching bioinformatic approach that he uses, I'll let him describe that to you. He has taken steps to do interventions and find drugs that would block certain classes of gene expression and perhaps promote a healthy aging. And also he has active collaborations with the JACs to do this. So he's part of our network here on the island. You'll hear more. You've already heard about the microscopy innovations we're trying to do here. Prayag, Marco, Pende are both authors on this paper looking at a new type of microscope, which enables you to look at live samples rapidly, efficiently, and really gain a lot of data on globally imaging. Jared Rollins is innovating in aging and how different compartments of the organism speak to each other in terms of regulating what proteins they make. And so he's innovating in new methods to visualize protein synthesis on a whole organism level. And he published this nice paper in Cell Reports Methods. And final example, not an exclusive list of what everyone's doing, but Jane Disney is really nationally recognized for her educational outreach and citizen science approaches, where she engages students and even high school students in collecting data. And she's been a real spearhead in basically characterizing water quality across the state of Maine in terms of arsenic content, and now an interest in PFAS. We've all heard about some of these forever chemicals. And so it's really by leveraging, the crowdsourcing of data acquisition that she's made these maps of the state of Maine in terms of water safety and changed people's practices, intervened in family life by getting people to put filters in their house. So it's not just us. We continue to draw the world to MD Ibl. Our visiting scientists publish nicely. A couple of papers. Jorg, who's here perhaps, has published a nice paper on using the zebrafish as a test model for nanoparticle delivery and also kidney function. Mark Seidel continues to run his classic courses here and is published nicely with his colleagues on gene expression in fish, and also the impact of basic science for clinical practice. And even people who've left. Sandra Riga left a couple of years ago, successfully got a job in Florida. She was part of our COBRI program, continues to cite Md Ibl as a source of support for her work. So we keep our community strong. And part of keeping our community strong is within the Md Ibl. We're a small place. There's only 11 of us right now. And so we really have to be more than the sum of our parts. We have to synergize with each other. And this will become essential as we think more about program projects, multi-PI grants. So here's a few examples of currently ongoing collaborations. Jane, Disney, and Jared Rollins are developing new ways to use C. elegans as sort of a canary in the coal mine for water quality and be able to detect things that may be faster, simpler, and more sensitive than some of the chemical analysis for environmental factors. James Godwin, Joel Graber, and Prag Mirawala are, as I mentioned, are pushing axolotl informatics, and they've applied for an R24 grant, and they'll reapply for that, basically to create a portal for researchers internationally to use the axolotl as a model for their favorite regenerative mechanism. Romain Madeleine and the Mirawala lab are working together to clear adult zebrafish. It's really amazing. With chemical modification, you can make the entire fish transparent and look at an entire fish in toto, get all of the structure of the fish for things like the vascular system or the nervous system. And so that collaboration is ongoing. Jane, Disney, and Jim Kaufman are working to better define arsenic toxicity using zebrafish as a model. My lab and Dustin's lab and Ron Corstagni from the JACS, who we work very closely with, who's an adjunct faculty member here, are all happen to be working on the same gene. Surprise, surprise. And so we'll hopefully be able to use our comparative approach to learn different aspects of function. I mentioned Sam Beck and James Godwin are working together on this idea of transcriptional noisiness and how some of these genes that are misregulated as we age actually promote inflammation. And this plays into James's expertise in the immune system. And so they have a collaborative funded project to move ahead on that. And finally, Herman's group and Eric's group, as you've heard, are pushing the envelope on this new model system, the African turquoise killifish for aging studies. And it might sound simple to grow fish in a tank, but I'll tell you, it's not a trivial business getting fish to mate, be healthy, and eliminate variables. And so establishing that here and expanding that here will make us a center for using that model for aging studies, which is practical. They only live 6 to 8 months. That's a time frame a postdoc can get an experiment done, as opposed to 5 years for zebrafish. So in terms of grant submissions, we've been very busy and active, and we thank Amy Pelletier for putting up with all of our late grants and workload that we give her. She's really been a key element in our ability to get these out the door. In the last 2 years, we've put out 32 grants, 10 of them awarded for a 31% success rate. And this sort of list below is the sort of variety of grants that we've applied for, mostly federal, but also local foundations. And we continue to beat the average for people applying to the NIH, which is about 20% success rate. So I think the state of our activity and success rate is strong here at Md Ibl. So looking forward, we currently have 8 grants pending, and just for thinking about the future. This is not set in stone. If we think about a 20% success rate, that could bring in almost $500,000 in annual support in the upcoming years. This is probably one of the most important slides for me, is how we're going to really leverage what we have into more and keep ourselves successful. And so clearly each one of us has to get an R01 level grant here, and we're working very hard at that for all of the investigators. We have a very active internal review process with very open, critical thinking about each other's projects. And I think it's a very strong scientific community. So I congratulate all of us on really being active in each other's grant activities. The acquisition of an R01 level grant is not just to fund our labs to do a project. It qualifies us to take part in multi-PI grants. So if we think about center grants and program projects, we all need to be R01 funded to participate. And when we think about those types of grants, we are focused on aging and regeneration here. So we do have a coherent synergy to apply for that. But if we can expand up to the 12 or 14 groups that Herman's talking about, it just gives us more leeway to put together different sets of investigators to grow our grant program and grow our scientific program. This year, we initiated the second R01 awards that Herman and I have talked about. The idea is that once someone successfully gets an R01 grant, more than likely they've got lots of scraps in the drawer that could be used for a second grant. And basically building on the work they've done and the crest of the wave they're riding, it's much easier to get a second grant once you've got the first one. So we give pilot seed funding of $75,000 a year for 2 years. And we're optimistic this will lead and leverage the first award into a second one. Herman mentioned our graduate program, which is the postdocs. This is our engine for the labs to generate this wonderful science that we do here. And so we're actively pursuing federal funding for that and foundation funding for our students. University of Maine has put together a new COBRI grant, which is exciting. It's focused on cell communication, environment, how cells sense their environment. And so Romaine Madeleine is a project leader on that grant proposal, looking at how the environment and signals impact muscle maintenance and basically forestall sarcopenia. So that's a really interesting project he's working on with that. And it got fairly, I won't disclose what I know all of it, but I think we're in the running. In terms of the upcoming years, we're looking to renew our COBRI, or actually it's a new grant for COBRI 3. And this would be primarily focused on our core facilities. So supporting the microscopy, bioinformatics, and animal cores. And the Embry grant will be a major renewal, and really want to congratulate Dustin and Jim Kaufman for doing such a great job on keeping that grant as vibrant and impactful as it has been. Just a brief aside on this COBRI, it's designed to really promote young careers, and we've been very successful in promoting, graduating 5 investigators shown here, Sandra Rieger, Jim Kaufman, Dustin Updike, Boot Yin, and Vicki Losick. And this past year, Sam Beck joined the club, and he is riding a great wave of bioinformatics and pushing the envelope on aging, as I've mentioned. And perhaps just as important as his success in leaving the COBRI is the lead author of his paper, Junyoung Lee, got a job at a prestigious university in Korea just this past week. So we're graduating people from their lab and seeding the world with smart people. So that's our game plan, and I think it will bring us credibility and maybe get some Korean students back over here. And our current COBRI PIs, James Godwin, Jared Rollins, and Prayag Mirawala are all actively applying for R01s, and they've been working very hard on that, and we're optimistic for them to do just as well as Sam and the rest of the crew. Okay, so you heard about the new imaging core. I'll throw in my 2 cents. Really, it's to create a center here that attracts people from the rest of the planet to come use technology, learn, interact, collaborate with us. And the innovation here is that this way of imaging preserves resolution, but accumulates a great scale of information. So you look at very low power, but very high resolution. And my analogy is it's like a Google map or something. You can look at the whole country, but then zoom in on Md Ibl and actually see the buildings here. And so in a sense, you can take these very large images here. An example from their website is the nervous system of a chick, I believe. Prayag, correct me if I'm wrong. Okay, so, but you can see the whole thing and get the, for instance, for a disease state or a mutant, you can see, you know, the whole picture of the deformities or the pathology, but then zoom in on a cell and get high resolution views and really get, it's an astonishing amount of data. So we're very excited about that, and we see future potential for having meetings here focused on this, bringing people here and enabling them with small grants to use the equipment and publish. And I think it'll be great for the Institute. In terms of bringing the world to Md Ibl, we've had very successful symposia over the last year. I organized this one on aging and stem cells, and we had 63 people attending online, really worldwide. The one benefit, and there's lots of downsides of Zoom, but the one benefit is you can invite someone from South Africa, you know, and they don't have to travel. So we had people from Egypt tuning into this symposium. And we basically focused on young PIs. We didn't have any established investigators really. 2 keynotes, but the idea is that the pandemic's really hurt young people a lot. And this was an effort to give them a platform. We also hosted the International African Turquoise Killifish Conference. Eric Rogers organized this last September. And that was a great success because this is really the ground floor for you. It reminds me of zebrafish in around 1996, you know, when people were still figuring out how to grow them, how to, you know, what's the genome, how do we do studies in them. But these have such great potential for aging studies that we're all very excited about that. Herman mentioned we hosted the MBMSS meeting, which is a great service to the state of Maine, and we had a great keynote speaker, Connie Sepko from Harvard. And upcoming, Eric is organizing, along with Ron Korstany, the iCarb course, and it's really a unique way for young investigators to get initiated in the field of aging and regeneration, but also to bring in lots of established leaders in the field and basically create a community here for 2 weeks around aging and regeneration. And then Jared is organizing the symposium that'll be put sort of in the middle of those 2 weeks, the mechanisms of cellular resilience, all focused on how cells respond to stress or less than perfect conditions, but how that impacts their ability to regenerate and the ability of the organism to have a longer lifespan. So we think that'll be an exciting way to bring new people here too. Jim just spoke about MDI Biosciences, but from the faculty point of view, it's a great outlet for a lot of the results we have that we wouldn't necessarily see continuing on in our labs. We see a lot of things, and we have to focus to get a paper done. But some things, serendipitous discoveries, can really have a powerful impact. So some of the drugs we use or treatments we use could affect our organisms. And we can feed that directly to Jim to exploit and to find a market and to enable that to not just be, you know, discarded with the random dust in the lab. Also, each of us has a unique, deep, vertical knowledge of our models. And we can impart that to Jim, and we can enable him to see new ways to use them for his vision of toxicology and other useful things. All right, so that's it pretty much. I thank you all for being here and for your support.
Well, thank you, Ian, very much. We have a couple of minutes for questions now before I hand back to Alan. Urgent questions to Jim, Ian? Yes, please.
Well, I just spoke to Jim about this, but it's worth thinking about. His slide of the Valley of Death, the drug development slide. There's another branch, and that is the branch of tool development, and which also has a commercial application. So that and it's a little more encouraging. The plain Valley of Death.
Yeah. I mean, you know, the Valley of Death is actually, we can talk about this during lunch, is what we want pharmaceutical companies to get to. I mean, we want them to demonstrate, don't go this way because your drug is dying. And but you're right. And we have talked about a lot of things. But this model development, I mean, we have been over the last, we introduced MDI Biosciences in March. And during the last couple of months, we have talked to a lot of pharmaceutical industries, to smaller biotech companies also. And we are learning and we are developing tools. I'll give you one very nice example. And this is an Md Ibl model to screen drugs for senescence or against senescence. So senolytic drugs, that's a big issue. When you think about it, how do you find out that a drug actually prevents you from aging? I mean, you can take it and then you wait. So you can actually do this in mice, but these are really tedious experiments. So how do you screen? This is a very serious question. How do you screen for a senolytic drug? And we have one approach, and I'm very proud. At the moment, it's the only approach. What we have found is that you take a zebrafish embryo, and when you have an aging cell, you inject it into the yolk sac. This is where the embryos feed from. And when the cells are aging, they release substances which we can then see in the zebrafish. And when we add a drug to that, which prevents these effects of the aging cells on the zebrafish, we see a reduction of the phenotypic change. We have patented this. At the moment, as I said, it's the only model worldwide which you can use to screen for senolytic drugs. So we have started to talk to drug companies about that. They're astonished because we have not published it yet, but the patent is in. So we'll see how things develop. And there are other models, and we could have filled another hour talking about other things which have developed at the lab and what has happened over the last year. So altogether, I mentioned this before, and you can see this from what Ian has presented and what Jim has presented. We had a very busy year, a very active year. I'm very proud of what has been achieved at Md Ibl, and let's hope that we sail into a bright future over the next year. And with that, Alan, it's really unfortunate. I mean, I'm not used to that. I speak into the void, and I hope that you can hear me. It's all yours.
Thank you. Well, many, many thanks to Herman, Jim, and Ian for their interesting and inspiring presentations. And I want to thank everyone in the audience today that's there in person or remotely for your continued commitment to the lab and for your support. Of course, that support is more important today than ever, given the ambitious plans that Herman has laid out. And for those of you that may be attending for the first time, please, we encourage you to get involved, to learn more about the important work the lab does, and of course, to support it. I think Peter Allen is in the auditorium, and Peter is stepping off the board. While he may be gone, he will certainly not be forgotten. And we want to thank him for not only his very long service on the board and his important contributions to the work of the lab, but also his chairmanship during a particularly challenging time. And so we acknowledge with gratitude his many, many contributions to Md Ibl and thank him for that. Okay. Everyone who's present is invited to lunch under the tent, and I'm very jealous that I can't be there. And during lunch, there's going to be a brief conversation with Dr. David Evans, who is the lab's resident historian. He's preparing to publish a new book featuring two of the lab's earliest scientists, Warren and Margaret Lewis, and he will talk about that. And then just a reminder that the trustees will convene at 1 o'clock in Dahlgren Hall for our board meeting. So again, thank you all for being there today. I hope you enjoyed the presentations and are excited about the future as we are. And with that, will someone make a motion to adjourn the annual meeting? Is there a second? Great, thank you very, very much.